Hello everyone, welcome to this video about jaundice pathophysiology. Jaundice is basically a yellow discoloration of the skin and the mucous membranes that occurs when bilirubin levels in the blood exceed 2.5 to 3 milligrams per deciliters. The best way to visualize this is if you look at the upper sclera of the patient where you'll find a yellow discoloration. There are three types of jaundice. There's prehepatic, hepatic, and posthepatic. We'll get into those later. Now, what's important to know is that bilirubin is a substance that's normally produced by the body, and the body also has a normal mechanism to which it can get rid of bilirubin. Any pathological process in either producing bilirubin or getting rid of bilirubin will lead to exceeding levels in the blood and thus jaundice. First, we need to understand the bilirubin pathway. Old red blood cells are broken down in the spleen by phagocytes that engulf and break them down. Heme, a part of hemoglobin, is broken down forming biliverdin that is then converted into bilirubin, and I mentioned in the slide that it's a hydrophobic molecule, so it can only be carried in the blood by albumin to the liver. In the hepatocytes, it's conjugated with glucuronic acid to form conjugated bilirubin, then secreted from the cell by multi-drug resistant protein. It has this name because it's associated with antibiotic resistance in bacteria. Conjugation is really important because now it's a soluble molecule that can dissolve in bile. Conjugated bilirubin in bile is then drained via the biliary tree into the small intestine. Intestinal bacteria then play an important role, first deconjugating bilirubin by removing glucuronic acid, then converting it into urobilinogen. From here, urobilinogen has two fates, either converted into stercobilin and released in the stool, giving it its brown color, or being reabsorbed back into the body where it's either reused by the liver in bile or converted by the kidney into urobilin and excreted in urine, giving urine its yellow color. So what is bile? I mentioned here it consists mainly of water that has two main components dissolved in it, bile acids formed of cholesterol and conjugated bilirubin. It's formed by liver cells, then descends down the biliary tree into the duodenum where it performs its main function. It emulsifies large fat droplets in food into smaller ones that can be absorbed, so it's essential for fat digestion, but it's also used to get rid of bilirubin. Just a quick recap on liver histology. In the picture we can see the hepatic globule. In the center is the central vein that drains into the hepatic vein, then IVC leading to the heart. At the sides we have the portal triad, consisting of bile ducts that drain into the right and left hepatic biliary ducts, the hepatic artery, and the portal vein. Between are hepatocytes consisting of three zones, zone 1, 2, and 3. Bilirubin is conjugated in the hepatocytes in the center at the centrilobular zone, zone 3. The conjugated bilirubin then moves peripherally towards the portal triad where it will drain into the bile ducts. The biliary tree starts from when the bile ducts all around the liver drain into the right and left intrahepatic bile ducts. These then leave the liver and so are called extrahepatic bile ducts. They join together to form the common hepatic bile duct. The cystic duct that drains the gallbladder opens into the common hepatic bile ducts to then form the common bile duct, which merges with the pancreatic duct to then empty at the second part of the duodenum at the ampulla of Vater that's guarded by the sphincter of Oddi. Now let's talk about the types of jaundice, starting with prehepatic jaundice. It's most commonly caused by hemolysis, which produces large amounts of bilirubin that needs to be conjugated. So the liver is overwhelmed and unconjugated bilirubin builds up in the blood, leading to indirect hyperbilirubinemia. The color of stool is normal as bilirubin is still conjugated, although not able to meet the high demands. So stercobilin is still produced, giving the stool its normal brown color. The color of urine will also be normal, as the unconjugated bilirubin in the blood can't be filtered by the kidney, so it won't end up in urine. However, we can find high levels of urobilinogen in urine, since large amounts of conjugated bilirubin is produced, then converted into large amounts of urobilinogen that's absorbed by the intestine and then excreted by the kidney. Now, hepatic jaundice is basically when you've got hepatocyte pathology leading to a problem in conjugating and secreting bilirubin. If it can't conjugate bilirubin, this leads to accumulation of unconjugated bilirubin in the blood, and it won't be able to secrete any of the conjugated bilirubin. So conjugated bilirubin will also accumulate in the blood. This leads to dark-colored urine, as conjugated bilirubin can be filtered by the glomerulus, and so it ends up in urine. Since we have both conjugated and unconjugated bilirubin in the blood, it's called mixed hyperbilirubinemia, direct and indirect. Finally, posthepatic jaundice is most commonly caused by obstruction at the biliary tree, mainly due to a gallbladder stone. So we can't get rid of the conjugated bilirubin, it builds up in the blood leading to direct hyperbilirubinemia. Since the bilirubin doesn't reach the intestine, it won't be converted into urobilinogen, then stercobilin, leading to pale stool. Since conjugated bilirubin is in the blood, it's filtered by the kidney into urine, leading to dark-colored urine. So we just have a table here to summarize the most important points regarding the three types of jaundice. Mainly we focus on the urine color and stool color when clinically examining a patient. Lastly, the types of hyperbilirubinemia. They depend on the levels of unconjugated and conjugated bilirubin. In indirect, around 80% of the accumulated bilirubin is unconjugated, while the conjugated bilirubin is less than 20%. This occurs in prehepatic jaundice that's mostly caused by hemolysis. In mixed, we have both conjugated and unconjugated. The levels of unconjugated are between 50 to 80%, while conjugated bilirubin is between 20 to 50%. In direct, it's mostly conjugated bilirubin in the blood. Unconjugated levels are less than 50%. To test which type we are dealing with, we use the Vandenberg test. A blood sample is taken and placed on a diazonium salt media, and we see a change in color upon interaction of the bilirubin with the diazonium. If there's an interaction right away, then it's conjugated bilirubin. If we need to add alcohol for the interaction to occur, then it's unconjugated. If the interaction occurs before and after adding alcohol, then it's mixed. Thank you for watching.